worry if you're not fast. If you're not fast enough with these with the QR code, I will show the same QR code at the end. But I also have put several papers up and lodged them with a QR code during the presentation if you want to follow up on any type of detail. Marcel has done a nice job of introducing myself. Here you see we are teaming up for CUT Europe, and we have a big team behind us which is making all of this possible. But beside the team, we have sponsors which I have to acknowledge. There's a lot of industry, but also a lot of philanthropic donors which are actually outperforming the industry contributions. And the work we're doing is sponsored by typical sources. And as always, if there's anybody missing a logo here, please contact me. I always find a space here. I also would like to mention a few conflicts of interest. In the pyrogen field I'm talking about today, I'm consulting companies. So please be aware of this. The patents I have held are expired, so this conflict at least is gone. So let's talk about pyrogens. And a lot of this pyrogen testing business actually started with this gentleman, Paul Ehrlich. Some of you might recall some 20 years ago, we had him on our German currency, the 200 D-Mark bill, because he won the Nobel Prize for a drug, Salvarsan, which was the first real treatment for syphilis, which was considered at the time the third plague. So really a very, very prominent worldwide threat of infections. And his drug was a miracle drug. You see down a patient, and after 11 days with Salvarsan, problem was resolved already to a large extent. Yeah. The problem with Salvarsan was it is not bioavailable. Actually, it's also unstable. It had to be dissolved and injected instantly. And they observed then quite frequently fever reactions of the patients when injecting this. And this made pyrogen testing a standard routine in the production of some medicines. The term itself was coined earlier by Burton Sanderson already in 1875 for an unknown substance producing fever. Pyrogenesis, fever or heat generating, fire generating, this was the term they used. And this is where a lot of these things started. The pyrogen test itself was first described in 1912 using rabbits and their fever reaction to find out whether something to be injected is positive. And quite notably, already in this very first paper, 1912, they noted that the rabbits did react to gram-negative bacteria very well, but not so much to gram-positive ones. I will come back to this in a while, because it's something which is plaguing us, I think, until now. And since the 40s, in every country in the world, large volume parenterals and sometimes all smaller ones like your flu vaccine every year is being tested for pyrogens. The next big step was actually in 1951 when Westphal and Lüderitz discovered what this pyrogen of gram-negative bacteria is, lipopolysaccharide. This was a very important contribution. If I put in LPS, lipopolysaccharide, or endotoxin, and I found more than 180,000 hits in PubMed alone. So you see, this is one of the best studied molecules which exist. And I once read, if you have a model and you're looking for an effect, throw some endotoxin into it, you probably will have fun. But this structure and this availability of a component has really shaped our understanding of innate immunity, of bacterial recognition, and is absolutely key. At the same time, gram-positive bacteria, those in yellow here, have no difference in fever in humans. They are as frequent in the clinics. About half of the patients show with gram-positive and gram-negative. And there is no reason to assume that they are producing less fever in humans. On the contrary, you can also not distinguish infections by the fever response. The gram-negative cell wall has this lipopolysaccharide, which is not found in gram-positive. I believe that lipoteichoic acids is actually their counterpart. This could be for another lecture. We have published very broadly about this. More than 100 publications came from my group on this topic. And I will not go into any level of detail. I just want to say we have shown, I think, quite convincingly, and now it is part of many textbooks, that lipoteichoic acids have all of the properties needed to be accounted as a gram-positive endotoxin, which is really important when we come to the testing opportunities. So the rabbit already fails a bit on the gram-positive side, but the one which was developed in the 60s, the Limulus assay, also known as the bacterial endotoxin test in pharmacopoeias, this assay is completely blind on this side. It is only capable of detecting gram-negative endotoxin, LPS. It is a very important assay, both with respect to safety of substances, especially of water, for pharmaceutical purposes, but it's also an important economical force. If you see this assessment from this year, here the pyrogen testing market is forecasted to be around $1.6 billion in 2025, and more than 90% of this is Limulus. So it's really big business. The assessment is, from my point of view, a little bit inflated, but in general, it is an enormous market, pyrogenicity testing. The rabbit test still is the gold standard for this reason, that the Limulus assay does not cover the full spectrum, but most of the testing nowadays is done with Limulus tests, which were introduced in the 80s in pharmacopoeias worldwide. The Limulus assay accounted in around 2000 for about 90% of pyrogen testing. And so you could ask, is the problem now solved? No, there's these 10%, and these 10% are actually quite substantial animal numbers. The Polyelec Institute calculated 300,000 to 400,000 rabbits worldwide around this time. But also the Limulus assay is a non-perfect replacement. Its restriction to endotoxin is one, but it's also disturbed by many substances. A lot of things you cannot test, very difficult to have plant preparations, or even if you filter something with a cellulose filter, you have a problem of false positive reactions, and does not at all reflect human potency effect, which is usually a bit downplayed, but there can be differences of a factor of 3 log orders in potency in the Limulus assay compared to humans. Interestingly, there is now, in more recent time, recombinant Limulus assays available. These recombinant assays have the enormous advantage that you don't have to bleed, as you saw here in this nice picture, to bleed the Limulus, which is done when they're coming in May to the beaches here on the East Coast of the US in order to get their blood, because about 15% of these Limulus do actually, up to 15% do die due to the procedure. But they also have, in this crude extract of blood, they do have a second pathway of activation, which is activated by beta-glucan, like cellulose, which is absolutely not pyrogenic in humans, but it's giving a positive signal. So the recombinant assays overcome this, and they are slowly taken up by pharmacopoeias worldwide, but I think there's a tremendous opportunity here. The dream, however, was to not use the immune system of a horseshoe crab, but the human fever reaction. And actually in 1981, Charles de Narello, a dear friend and colleague, he was for 10 years the most cited author in the life sciences. He discovered the molecule which is key for our understanding of fever today, which is interleukin-1. This interleukin-1 is released by activated monocyte macrophages mainly, and then it initiates the cascade of producing fever. Already in 1984, he suggested that we could humanize, based on his molecule, pyrogenicity testing, because he showed that there is certain growth factors which we cannot really control with rabbits or the Limulus assay. And he showed that this worked out by using peripheral blood mononuclear cells, so the primary cells of the fever reaction, including the monocytes. But he still had to use rabbits as the detection system. So he had his cell culture produce interleukin-1, and the interleukin-1 then was injected into rabbits via fever reaction. It was only 5 years later that Steve Poole came up with the idea to use an ELISA instead, which had become prominent in the 80s. And he suggested to use the same cell system and interleukin-6 ELISAs. This was the state of the art when we came in a few years later. Albrecht Wendel, my mentor at the time, and myself, around this photo is around this time. So you can see we came up with the idea to simply use whole blood, because isolating these mononuclear cells is something which is quite difficult, demanding, especially for the control laboratories, which are typically doing pyrogen testing. And we settled on interleukin-1 as the primary cytokine. So this was our starting point. Parallel to all of these developments, there were a few people who suggested to use monocytic cell lines. And I brought all of them together. You see here the main consortium to then carry out a validation study, which lasted from 2000 to 2002. So here are the individuals. I cannot name them all, but this was the consortium actually to carry out the validation study of 6 in vitro tests. And we were funded by the European Commission with about $3 million. And it took then until 2006 for the advisory committee of the European Center for Validation of Alternative Methods, which I had the privilege to lead at the time, to acknowledge that 4 of them actually were considered to be validated tests. And these were the whole blood assays, the assay based on the peripheral blood mononuclear cells. And we had one of the cell line assays who failed completely pre-validation, but it made it suddenly in the validation phase, the assay on monomax cells. So these 4 assay variants, it was also a prior variant of our whole blood assay. So these 4 assays were considered validated from then. What was quite interesting in this context, we analyzed our point of reference, the rabbit assay. And what you can see here on this graph is in the Paul Ehrlich Institute, one of the most sensitive strains of rabbits was used, and they're considered the reference laboratory. 171 rabbits injected with endotoxin were studied, and you can see the enormous differences in responses. So that's not a good assay, because this is the distinction between positive and negative. So you can see you can very easily, with the small numbers of animals, just picking 3, you can have false negative results, even at relatively high endotoxin concentrations. And we set up the validation study where we used 13 different drugs, and the rabbit would have failed them. Yeah? Already based on these sensitivity data, it would be only 58% specific and 88% sensitive in the setup, and without any disturbance by the drugs themselves. The validation is published long ago. There's also additional work done comparing our whole blood assay directly with rabbit assay testing by the Paul Ehrlich Institute, but also work carried out in Cuba. You can see these studies here. So this is all history. What was also interesting, 2 of the participating labs, national control reference laboratories, carried out Limulus assays. Again, highest standard Limulus assays on the same samples. And what you can see is that the Limulus assay, the bacterial endotoxin test, would have failed this validation study in both of these labs. So it shows that there is really an advantage of these novel methodologies. In the US, unfortunately, different to Europe, the assays were not accepted as pyrogen tests, but they were only accepted as endotoxin tests, so only to replace Limulus testing. And this led us, in coordination with FDA, to submit a big dossier, which was also published, on accumulating the evidence for non-endotoxin pyrogens being covered. And despite our agreements, it has never been discussed. There was never a reaction by the validation body and the FDA on this dossier. But there's essentially no doubt that all of the monocyte activation tests, as we call them now, MAT, are capable of detecting exactly the non-endotoxin pyrogens relevant to humans. Interestingly, as I mentioned earlier, the rabbit has some failure in detecting gram-positives. And this was a study my PhD student at the time, Stefanie Schindler, carried out, where we simply did a whole blood assay with rabbit blood. And as you can see here very nicely, while human blood was capable of detecting here lipothiuric acid from Bacillus subtilis, a gram-positive, rabbits essentially failed to do so. And we have seen so also later in some in vivo experiments, there is a clear distinction in the immune system, and it might very well be that our complete emphasis on lipopolysaccharides came from the very beginning that the rabbit is one-eye blind for these type of pyrogens. You can compare the different approaches of cell lines, of PBMC, of whole blood, of cryopreserved blood. They all have advantages and disadvantages. I'm very much convinced that still the whole blood assay has some advantages. That's the assay we invented, which was validated, became commercially available at the moment through Merck Millipore as PyroDetect, and has been accepted in the different pharmacopoeias. I twice summarized my experiences on the validation process. So what I'm discussing today, very superficially, you can read in these two articles in much more detail. The advantages I see are those which all monocyte activation tests have over the rabbit. The rabbit has never been validated. It is human test systems with their reaction, for example, to gram-positives. We are dealing with quantitative assays. The rabbit is not quantitative. We have positive, negative controls included in the experiment, no animal use, and it is considerably cheaper, at least tenfold. It has, from my point of view, also tremendous advantages, all MATs over the Limulus assay. They're not restricted to LPS. They reflect potency in humans. There's much less interference, so substance not testable in the Limulus assay can be tested, and there's no animal suffering, no bleeding and death of these. From my point of view, there's also advantages in comparison to the PBMC variant, the main competitor, let's say. You avoid all of the preparation artifacts. We have a validated cryopreserved blood version. You can do incubations in a simple thermoblock, no cell culture required. There's direct comparisons to the rabbit available. It's much more data on non-endotoxin pyrogens. Human plasma, which is very important for recognition of many of these factors, is present, does not have to be added. It is more products validated. It's a cell suspension, which has big advantages because you can dip a medical device into this suspension or test on filters, which is very difficult with adherent mononuclear cells. And there's certain variants that we published which increase about 100-fold sensitivity. And there's also clear advantages of both PBMC and whole blood over cell lines, full validation, no cell culture. They're genetically complete. Many of these cell lines miss 10–20% of their genome. You would have to show for each and every pyrogen that the entire machinery is there at a given time, as they're very genetically unstable. And you see this also on the level of validation. We have an incomplete picture except for the whole blood assays here. Now, let's talk about the success story. We had, unfortunately, a time of 7 years where the EU was not publishing data on animal use. This changed in the beginning of 2020. This is a paper analyzing this, and the big story was that the enormous number of animals used in Europe has dramatically gone down by about 80% in the last year covered, 2017. And for comparison, in 2005, the starting point here, about 100,000 animals were used for all testing of chemicals, or the same for all testing of pesticides. So we're really talking about an enormous consumption of animals. Only about 5,000 animals were used per year for skin and eye irritation in rabbits. Yeah? So this is really a big thing. And the big news of last year was that the European Pharmacopoeia announced in 5 years, all rabbit testing will be done. I would like to end with mentioning a few applications of the assays, which are missed opportunities from my point of view, because these are not just replacing animal testing. Like many alternative methods, they are enabling technologies. One thing is air monitoring. Pyrogens inhaled in air, for example, in stables, in recycling plants, and in others. There's many areas can be monitored with these type of filter systems, and we have published on this. You see a few publications here, but there's nobody really following up. We do some work on childhood asthma at the moment, but that's it. Immunotoxicity. We have pre-validated a variant of the assay as an immunotoxicity assay, and only most recently there's some interest again into this because it seems to work simply. It has been also promoted, some of these assays now, as cytokine storm assays to test biologicals. So there is immunotoxicity really is interestingly covered with monocyte activation tests. Another largely missed opportunity is medical device testing. This was one of the really critical experiments we did in this area on the graph. What you can see is titanium, which was manually handled by people. And then we had a strong signal because of the pyrogens attached. And we hardly got rid of this. It took us 300 degrees Celsius in 5 hours to really remove this again. So on the surface of these medical devices, there can be enormous pyrogen loads just from the manual handling. The last area I would like to touch on is blood transfusion. So while every saline solution you get injected has been tested for pyrogens as a batch, we do in the US alone 30 million blood transfusions, and none of them has been tested for pyrogenic contaminations. And it has been calculated that about 1,000 fatalities occur in the US alone based on microbiological contamination of blood products which are not being tested for this. There's an enormous opportunity here again. So what are the lessons learned? I think it is an excellent example that alternative methods can outperform animal tests. They can be better. And they can be so if they're based on mechanisms and human cells, so they're relevant. I think it was important for the success of these assays that they also found commercialization, because it was the pressure of companies bringing them to the market, standardizing them, financing these long journey for pyrogen testing with alternatives. 15 years to acceptance and 30 years for full implementation in Europe is too long. We have not yet reached this in America, Japan, Brazil, and others. There is lack of enforcement of implementation of successful methods. We have no law. Nobody is penalizing you. They are enabling technologies. They allow us to do things which go beyond what the animal test could do. It is all only possible if there is a collaboration between the public and the private sector, if there is pressure on all of the actors, if the regulators work together with the industry and the academics to get this done. And there's an enormous importance for international harmonization. I'd like to announce that we are planning now for a workshop towards animal-free pyrogen testing in America, because unfortunately, here we are far, far behind the European developments. And my last slide, I have to thank many people. More than 60 were critically involved. This is what I listed at the time. I cannot name and mention them. Many dear friends who have made this journey possible. And I would hope that we have time for a few questions.
Good, perfect, Thomas. Thanks a lot. It's a wonderful talk. It was even exciting for me. Also, I've heard the topic many times and I couldn't stop listening. So I hope it was equally interesting for others. And of course, there are opportunities for questions.
Maybe we can start the poll. So
yeah,
then
so everybody gets warm, everybody gets their fingers on the keyboard so you can type in questions. So Georgia, let's start with some very easy first two overview questions.
Okay. So I started, I think for the panelists, sorry, I think you cannot see it.
I can see it.
Ah, you can see it, okay, good.
So you have to give instructions perhaps, or is it clear?
So I see that someone has started to vote. The second question is long, so let's give it some time. And in the meanwhile, I can remind you to use the Q&A function. to ask the question, so it's easier for us. So I see that more people are voting. Shall we give more minutes? Or I'll...
Well, questions are not so difficult. It's just for getting warm.
Okay, then I finish the poll, be fast. And I share the results.
This is amazing. So we have some experienced users around. Good. So I guess we will also get a mixed bag of questions. Some new to the field, some coming in. And then the other one was, I give a brief feedback just on the outcome. So now this talk addressed, of course, this big issue of, you know, how is transfer done between labs and application. And many might not realize. So there's a big agreement on this and a partial agreement altogether going towards 90% or higher. So, okay, look into your shelves, look into your experiments, and always think of how that could be done. So Thomas did this during his PhD thesis, and it ended up quite successfully. Good, but then let's go on. Like you can ask questions, I guess, by vocally or by typing.
Yeah, so the first one, I will also put them live. What are your thoughts on donor to donor variability from standardization point of view for the whole blood map?
That's a very important one. Actually, most differences in reaction with respect to the amount of cytokine produced comes from the difference in monocyte counts. So you already standardize a lot by calculating the per monocyte. What is extremely high conserved, and this is what is actually being used for these assays, is the set point where it starts, the point of departure for stimulation. And I think you can imagine this is extremely important for the body. We live with about 1 kilogram of gram-negative and 1 kilogram of gram-positive bacteria in our gut. So this is 50 grams of endotoxin alone. So this could suffice to kill 1 million people by injection or produce fever in 1 billion people. So your body has permanently to control that there is no influx into the body. And this is what these reactions do. If you would overreact, be more sensitive than others, then you would have permanently inflammation in your gastrointestinal system. If you're too little reacting, you would be invaded by bacteria. And this is controlling why the set point where this starts is extremely conserved between individuals. And we have done up to 152 people in a single incubation, or actually 2-day incubation, to control the reactivity towards endotoxins. Yeah? So it is not really a problem. For these assays, it only means how high goes the curve, but the point of departure is critical. And then using an endotoxin standard curve, you always have a dynamic range, which is pretty much the same.
Okay, we have another question. Is there an objective reason why Europe implemented it, but not Japan and USA? What about OECD?
The OECD plays no role here. This is not a test used for chemicals. It is a test which is used mainly in the quality control, the batch quality control of pharmaceuticals. And this means the pharmacopoeias of the world are necessary. And I have to say that we, first of all, had the European Pharmacopoeia as an associate partner already in the validation study. All of these developments took actually place in Europe. These were European assays which were pushed. the validation was run through the European Center for the Validation of Alternative Methods. And then it was really, again, the Pharmacopoeia with their working group who pushed the agenda. And there's people, especially from the Paul-Ehrlich Institute, Thomas Montag-Lessing, Ingo Spreitzer, then following, who made this happen. We are lacking such pioneers, such champions on the US side. On the contrary, you could say all of the Limulus industry is US, or most of it is US. And for this reason, there's also sometimes non-scientific barriers for the acceptance of these methodologies, which we have encountered over these 25, 30 years.
Mm-hmm. We have indeed also a question from Ingo Spreitzer. So, dear Thomas, do you think the FDA Modernization Act, if accepted, will finally bring the MIT acceptance to the US?
Well, what I see is that the FDA is in general extremely positive towards alternative methods in recent time. The leadership of FDA has really been extremely supportive on various fronts, not everywhere, but what we have seen, for example, that they have now introduced some fast-track qualification procedures for alternative approaches. We have seen that the FDA just last year formed an inter-center working group on alternative methods. So all of the 6 FDA centers work together here, and we have a very nice working relationship with them. I'm carefully optimistic. I'm not sure whether the Modernization Act itself will be so meaningful. I think it depends here really again on the champions in moving the agenda. The problem at the moment, let's say the hesitance here on the US side is more on the side of the US Pharmacopoeia, but also they have, after all these years, 2 years ago, had the first workshop on the novel pyrogen tests. So everything is moving. It is just not as fast without champions. Yeah.
Mm-hmm.
Perhaps I pick up an easy question, but a good one. Is the number of rabbits referring to tests in rabbits, or is it the number of rabbits really being used and killed in the end?
These were the number of rabbits. The tests are, you have to divide by 3 to 6. The typical setup is a 3-animal test, and if there's an unclear result or repetition, so it's somewhere probably by 3.5 or so, you would have to divide to find the number of samples tested. But I would say there's tons of things we would love to study, but which are not being studied in pyrogen tests because the animal is simply too difficult, too costly to handle. I think with the acceptance of a real pyrogen test, there will be a lot of questions which will be answered in the near future, where the full pyrogen spectrum has to be assessed. Yeah. So it's not only about replacing the rabbit test.
Okay, perhaps another question that fits with this, and you mentioned the problem of masking of endotoxins. Is this overcome by the MAT?
To the extent we have studied it, yes. So this is a very interesting one. The main reason for pyrogens not being detectable is that they absorb to materials. This is a big problem with proteins, especially. The Limulus assay does not catch them. And what is dangerous actually is that the typical procedure for the Limulus assay is to demonstrate that you can recover endotoxin by freshly adding it and then measuring it. If you give it hours or days to really absorb to the material, it vanishes. That's what's called masking. And for many of these materials like albumin, it was clearly shown that the Limulus assay does not detect it anymore, while it is detectable with the MAT. But there's so many materials that I cannot speak for each and every one. It would need a product-specific validation, but I think that a lot of materials become testable as our own experience says.
I guess this is important for especially recombinant human proteins like blood factors and so on, which probably are a huge problem in the rabbit.
They are, and you should also not underestimate that the necessary volumes are a big point here. The rabbit is only about 1/10 in sensitivity towards humans. So we are really much more sensitive to pyrogens, but this is compensated by injecting 10 times more into the rabbit. But for extremely expensive stuff like blood factors, coagulation factors, it is painful to inject into a rabbit. Yeah, if you know what it did cost to bring so many donors together to get the respective materials. So also the fact that you can miniaturize this, you can work with a droplet of blood in a 96 well plate and run the alternative assays. That's a big advantage too. Yeah.
Good. And you also mentioned the monomax cells that failed the pre-validation. Did they enter the pharmacopoeia, and is that understandable?
Yes, they are listed as an alternative method. There's also commercial variants of the monomax 6 cells. I would say, my personal view on this is I've seen how variable these assays are. So we had 3 laboratories in the validation study carrying out the monomax test, and for 2 years we had no consistent results between the 3 labs. And we were always discussing, do we drop them? Do we keep them? And we did not want to lose our colleagues from the Netherlands in case, which were the lead laboratory for this. And we kept them in. And suddenly, like a miracle, in the last and final study, they worked in the big validation study in all 3 labs, while the THP1 cell line assays suddenly failed. They had done the pre-validation well. And I think this hints to the general problem of cell lines, especially when freshly cultured. This is obviously different if you can have ready-to-use cells. You take from the fridge, instantly use them. Some of the developments which were more recent, but they have never been formally validated and have not undergone controls. And I'm also not aware of any broader studies showing to which non-endotoxin pyrogens the monomax cells actually do react. So they come with some caveats, but they have obviously advantages with respect to infection protection of workers, some concerns. Another concern you always have with a cell line is this is one donor. For the blood cell assays, we request multiple donors in order to exclude an unusual reaction. And as you don't know what is the pyrogen in the sample, you cannot do this with monomax. You would need several donors, monomax cells, and this is not existing. Yeah.
There is another question. Are there any development towards real-time detection of pyrogenicity, for instance, continuous flow systems?
I'm not aware of anything based on these novel assays. The shortest incubation periods I have seen are in the 1 hour range. If you're moving from cytokine production to transcription assays. So there's always a delay. You cannot measure it instantly. There is some devices to do fast Limulus tests. So these type of things are available. I would say they are coming close to real-time opportunities, really. No? Mm-hmm.
And you mentioned a lot of material, and very interesting also the solid material in contact with blood. Partially solid material could be small particles or nanoparticles. Is there any experience on the testing?
Yeah, actually, this is a very important one. I skipped this in my presentation due to time. Nanoparticles have tremendous surfaces. Yeah? If you have a 10 nanometer nanoparticle, 1 milliliter, this is the cup of the top of your tip of your little finger, your pinky. Yeah, 10 nanometer particles would have the surface of a soccer field. Yeah? So you can imagine how much they can absorb, and this includes endotoxin. And what has been shown is that nanoparticles, for this reason, often mask endotoxin in the Limulus assay. But it has also been shown that the whole blood assay can handle some of them. Again, there's not broad characterization, so we don't know whether this is a general statement, but there's tremendous potential for the increasing number of nanoparticles being used to control them for such contaminations. And if you just imagine how many are injected for medical purposes, for radiography and so as contrast substance, I think there's a tremendous opportunity to control nanoparticles with these assays.
So could one go on a step further with the question that even live bacteria might be masked by the high surface? So the sterility question as such?
It is not clear, but what's quite interesting, the MAT are capable of detecting live bacteria as well as they detect the pyrogens, so their parts. So we have done this, for example, in the context of this blood transfusion stuff. So you would get, it's not nice to say for cut two birds with one stone, but it is actually almost the fastest microbiological test for the presence of bacteria as well, as it does not distinguish between live and dead. Where the masking can take place, I could imagine. Actually, albumin is one of the best binding components for endotoxins. So I could very well imagine that the coating of bacteria with albumin can be a problem, which is impairing some reactions. But I'm improvising here. I've not seen this. But for example, lipopolysaccharide makes up about 70% of the surface of a gram-negative bacterium. So it's really it is the so-known O antigen. That's what antibodies are formed against. These are the carbohydrates on the surface of the bacteria. So it's really I could very well imagine that there is some masking effects taking place.
Good. So as much as I can see, we are running dry on questions on the talk on endotoxin assay, pyrogenicity, technology transfer in this area.
I will try to answer, which I see here in the chat. Still, I will try to answer in writing. Yeah.
Yeah.
There's I see here quick ones. Horseshoe crab, are they dying? As I said, up to 15% have been calculated. 5 to 10, usually most of the producers say. So there's a discrepancy. It's difficult to follow up when they are thrown back into the water by fishermen. I had some users' problems with cryoblood. That's a cool chain problem. Yeah? So you really have to maintain very good cool chain, and you really need -80, minimum -70 degree. There have been some developments of variants which are better, but the cool chain is a problem, yeah. Especially if it's not an extremely well-trained laboratory, and we have seen it's difficult to cross borders with these. In the experienced labs, it was not a problem at all. Difference between the US regulators.
I think we had this already.
We had this covered, yeah. Yeah, Kirk is asking about a question which is different. That's a discussion we are having about testing of the new vaccines. I'm happy to continue a discussion, but not in this forum. It's not related to the talk.
Yeah.
And then what do we have here? Based on regulated environment, do not adopt the best impact class assay unless it's entered in the pharmacopoeia. Yeah, that's an important one. I agree fully. It is a key step to have them in pharmacopoeia. And then the next step is to have pressure on the industry by the regulators or outside animal welfare groups and others that things are being implemented. Because especially in the established drugs, nobody wants to change something which suddenly might lead to some batches to fail. It is a key step here, yeah.
There is a last one entering. How about TAL, Tachypleus amebocyte lysate?
That's an interesting one. This is a very closely related crab to Limulus polyphemus, the crab from on the east coast of the US. It has a very similar reaction. The Japanese production and also Chinese production is done with this because they have only this species. I'm not aware of major differences between the two. So it's just, let's say, what source you use. Fun fact, I learned at some point, because Cuba was boycotted and did not have access to pyrogen tests, that they actually used lobster blood, which is also capable because lobster they had in order to produce a similar hemolymph assay. But I cannot talk either to the lobster blood assays, how good they are, but it seems simply to be a defense reaction of a broader variety of species. And it happens to be a Limulus, which is the most prominent one. Yeah.
Good. So I think we also arrive within the time frame. Like we also want to keep this short and interesting for everybody. I think everybody is invited to compete on some questions on the contents of the talk. I think something was taken up again in the discussion. Perhaps it helps for answering. And I leave this to Georgia now.